Record. Ugh. All righty. So we ended, oh, I don't have it up there. We ended talking about the genetic code. Bring the chat up. Down here. So this is the last slide I think I talked about. And I'm not going to have you guys actually determine like what amino acid. You don't have to be able to read this for this class, but just take note that there's essentially
I can't hear anything.
all the different 3-letter codes that make up the 20 different amino acids. So this simple table is basically, in a sense, what makes us who we are in a lot of ways, because proteins are a huge part of our functionality, and this codes for proteins eventually. So it codes for the individual amino acids that will make up the protein. So that is the genetic code. That's where I ended. Now we're going to get into the process of moving from essentially DNA into a protein. So that'll include transcription and translation. Hopefully some of this is a review for you guys. I am going to kind of hit on the main points like I've been doing for some of this sort of more general bio review information. So if you're totally lost on what the heck transcription and translation are after I do this, definitely go to your textbook or come meet with me and I can explain some of the missing pieces. I tried to give you the information you need without cutting out so much information that it doesn't make sense. So hopefully this works. Oh. Can other people? Oh, okay. Never mind, you can hear me. Okay, good. All right, so protein synthesis. This is the whole kind of the goal, a huge goal of many cells, of most of the cells in our body is to make a protein. The process through which that happens, I have sort of outlined down here. It's broken into 2 different steps, transcription and translation. So transcription is where the info from the DNA, which is where all of your hereditary information is held in the nucleus of your cells. Information from the DNA is coded into an RNA, which is messenger RNA. So DNA to mRNA is transcription. That messenger RNA is then translated into a protein. So translation takes that information that was coded into that mRNA strand and then makes it into an amino acid chain and eventually a functional protein. So I'm going to talk about these 2 activities, and that's really all we'll cover today. So first, transcription. This is where messenger RNA, a single-stranded molecule, mRNA, is made and processed based on what the DNA code is. So there's complementary base pairing. I talked about the base pairs that line up on Monday. So let's see, if we're talking about DNA to RNA, it's going to be A to U, or if there's a T in DNA. So A and T / U, depends on whether we're talking about DNA or RNA, whether we're doing T or a U, and then C and G. So thymine, T, is only in DNA. So when you're making the mRNA, if there's an A on the DNA side, it matches up with a U on the RNA side. And then C and G stay the same. If there's a T on the DNA side, that's just an A on the RNA side, right? So the complementary base pairing is really important, and we're going to continue to complementary base pair as we move into translation as well. So those are the base pairing rules. So you want to ingrain those into your brain and know whether you're talking about RNA or DNA. And that's going to determine whether it's a T or a U. You're basically only going to add U's because we're not talking about replication of DNA. So we're talking about making RNA. So we'll mostly be talking about uracil instead of thymine in these cases. So then after that complementary base pairing has happened, so this mRNA strand has been built based on complementary base pairing from the DNA strand. Some processing has to happen. So a couple of caps are added onto the end of the molecule, and then some parts are spliced out or cut out. I'll talk about that in a minute. And then this is all happening in the nucleus. So the DNA is in the nucleus, right? So transcription has to happen in the nucleus where the DNA is. That messenger RNA molecule that's built off of the DNA code then has to move from the nucleus into the cytoplasm. So it moves out of the nucleus through nuclear pores into the cytoplasm, which is where the second step, translation, happens. Okay, so those are the main pieces. I'm going to talk about kind of the individual steps here and how it works. After another quick overview, I always forget I have this slide here. So to give you sort of an image, I know that's a very busy image over on the right there of what's happening. Transcription happening in the nucleus, you have DNA, the double helix of DNA, and then being built off of that is this mRNA transcript, the single-stranded transcript. Let me move you guys over here. There we go. That mRNA transcript then leaves the nucleus. So here, the purple thing is the nucleus. It's leaving the nucleus and going to a ribosome, which is where the amino acid chain is built, and that is translation. We have two different locations here. All right. So I repeat myself a lot, mostly because I want you guys to, I don't want it to go past your, past you noticing it. So mRNA made from DNA, that is transcription. Transcription is going from DNA to make this mRNA molecule. The main enzyme that does this, really the kind of heavy lifter in all of this transcription process, is called RNA polymerase. So RNA polymerase is essentially what's making the new RNA strand, that mRNA strand. It's adding the complementary base pairs based on what's in the DNA. Yeah, and it makes that based on, it's called Chargaff's rules. You don't have to know his name. It's a guy that came up with, he discovered the fact that A and U are going to bond together, and then C and G are going to bond together if we're talking about RNA. So he came up with the base pairing, or he discovered that base pairing rule. So it's just a simple matching of bases. So based on what's on the DNA, that the complementary base will be added to the growing mRNA strand. In this video, it goes into, there's a few details you don't need to know, but I'm going to show you this video here. I guess the easiest thing to do, since half of you guys are on Zoom, this is going to take extra time, but that's okay. I'm going to show you guys here. I'm just going to put you in front of the screen. All right. So it's not an exciting video, but it gives you a good vision of what is happening with transcription, I think.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Okay, so you don't need to know about the promoter and terminator or the triple phosphate molecule, but I think that gives a good image, at least, of how RNA polymerase is basically just reading the DNA and then adding the complementary bases. All right, this is probably going to make this play.
A structure.
Okay.
A structure.
I don't know why it does that every time. Okay, so the complementary bases are added. That single-stranded mRNA is built. After that mRNA is built, it's not really mature yet, so it's not ready to be translated. So a few more things have to happen, and this post-transcriptional processing is really important. So what you have after that initial transcript is built is called pre-mRNA. It's just immature, is what they call it. It's not ready to be translated into a protein yet. So there's certain segments that need to be cut out, and then caps need to be added onto either end of it to protect it as it leaves the nucleus, basically. So the exons are the important parts. That's the red chunks here that you see. Those are going to be basically connected, and the introns, which are in between all of those, are going to be spliced out and removed. So they're non-coding parts of the immature pre-mRNA. We don't need them. They're useful for other stuff, so they're still important, but as far as making a protein, they're not useful. So introns you can think of as in the way, so they get taken out. Introns get spliced out, so now you just have this nice sequence of exons, this red unbroken coding sequence here at the end, and then the caps at the very ends, like I mentioned, are just for protection as this strand, this molecule moves from the inside of the nucleus to outside. So it's a little bit dangerous for the mRNA to move from inside the nucleus through the pore and then out into the cytoplasm. The ends can often get damaged. So we don't want the coding sequence to get damaged. So these caps are added on to either end that if they get damaged, it doesn't really matter. So we have the 5' cap. Don't worry about 5', 3'. It's just, they're two different ends. But you have a 5' cap and then a 3', what's called a poly A tail. Just means there's a bunch of adenines. So that nucleotide adenine is added on to the end. So yeah, that's for protection. And then you have this unbroken coding sequence with this protective cap on either end. So now it can move out of the nucleus and into the cytoplasm. Good? Okay. So now we're moving into the cytoplasm and onto translation. This all happens at ribosomes. So you guys know what ribosomes do. They're really important in protein formation, and this is why. This is where that mRNA molecule goes to. They go to the ribosome, and that code that's on the messenger RNA is then read by the ribosome in order to put a bunch of amino acids together to make an amino acid sequence, which will eventually become a functional protein. So that's the process of translation, and I'll show you what that looks like in images and then a brief video again in just a minute. This is where the other two types of RNA come in. So I mentioned there were 3 types. Transcription just involves the messenger RNA. It's bringing the message from the DNA now to the ribosome. RNA down here, that's just what makes up ribosomes, ribosomal RNA. That's all you need to know about the rRNA. The transfer RNA is what's bringing in the amino acids. So we need to build an amino acid chain. The transfer RNA, tRNA, is bringing each of those amino acids to the ribosome. It's bringing the correct amino acid based on the mRNA code. So we're going to have complementary base pairing again between the mRNA and the tRNA. So they have to have complementary codons, basically. And that will allow the correct tRNA with the correct amino acid based on this code and the mRNA to be brought into the ribosome. All right, so what does that look like? It's kind of hard to imagine. So on the right here, we have a ribosome with the 2 different kinds of RNA. Well, I guess all 3. The ribosomal RNA is the ribosome. The mRNA and the tRNA along with the amino acids being brought in. So I'm going to show you what this is so you guys can see okay. This teal colored thing is the ribosome. So here we have the ribosome, that teal colored blue thing. This is where all of translation is happening. This reddish pinkish strand with these letters on top are the mRNA. That's what we made in transcription. So we have the mRNA strand here. These weirdly shaped molecules are the tRNA. So they're really weirdly folded strands of RNA, basically. Each tRNA has a very specific amino acid that it grabs onto based on its anticodon on the tRNA. So here we have, for you guys on Zoom, this is the anticodon, which is part of the tRNA. That will determine what amino acid is attached to that tRNA. That anticodon has to complementary base pair with the codon, which is on the mRNA. So we have U and A, C and G, A and U. So we have that, those base pairing rules are still the same between mRNA and tRNA. And that's the only way you'll have the correct tRNA coming in with the correct amino acid, because we need a very specific sequence of amino acids to build the right protein. So again, the codon is the 3-letter sequences on the mRNA. The anticodon is on the tRNA, the 3-letter sequences on each tRNA. So the ribosome basically pulls this mRNA molecule through it, and it reads the next sequence of 3 letters, so the next codon on down the line. So this mRNA molecule will be pulled through, and each of the next codons will be read 3 letters at a time to bring in the right amino acid to add on to this growing polypeptide chain or amino acid chain. This is the amino acid chain, which will eventually be our protein. Okay, and I think I have a video next to kind of show you what that looks like in action. Here we go.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies.
You don't have to worry about the EP and the A site. Just understand the fact that tRNA is going to bring in those correct amino acids to the ribosome.
The P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
All right, so that is translation in action. Super exciting. It's actually pretty amazing that all the thousands of proteins that our body makes are based off of just 20 amino acids, and all of those are based off of just these 3 base pairs at a time. It seems amazingly simple for the complex level of protein development that we have. All right. Okay. And then, similar to after transcription, with the mRNA processing, a little bit of processing has to happen after translation. That happens at the Golgi complex. So remember, the Golgi complex is sort of the packaging, processing, and shipping area of the cell. That's the organelle that does all the packaging and shipping off. So a few modifications might happen to the protein, and then it is put into these Golgi vesicles and is sent wherever it needs to go. So after it's at the ribosome, goes to the Golgi apparatus or the Golgi body, and is kind of finished up and then sent to either another part of the cell within the same cell, or is exported from the cell to go somewhere else. So the Golgi plays a huge part in this kind of post-translational processing. Okay, I think that's really all I'm covering about the details of transcription and translation. This is kind of a nice summary. If you can follow what this diagram is showing, you have a pretty good understanding of how the process works, moving from the DNA all the way down to the eventual polypeptide chain at least. So we start out with the DNA. That has these codons on it, right? So these 3-letter chunks. The mRNA is built off of that with the complementary base pairs. And then this weirdly shaped molecule, this is the tRNA, that's going to bring in the correct amino acids based off of these codons. And then those amino acids will be bonded together into this amino acid chain. That's in a nutshell what's happening in transcription and translation. Any questions about that? I know that was kind of a quick run through, but hopefully it mostly made sense. Good, okay. And just one more thing I wanted to point out. So this is the last slide I'll go over. This is the last slide you're responsible for the exam. The fact that genes aren't always being transcribed and translated. So gene regulation is actually a really active area of research in biology right now, figuring out when genes are activated and when they aren't. A lot of times it has to do with formation of different kinds of cancers. So if a gene goes unregulated and it just keeps producing a protein or stops producing a protein, basically it can cause cancer in addition to a number of other sort of disorders. So genes can be turned on and off, and this sort of oversimplifies it as like a light switch. But ideally, in a correctly functioning human cell, the gene is only turned on when that protein is needed, right? So a good example of this is if there is alcohol in your blood, the liver cells will turn on a gene to make alcohol dehydrogenase, which breaks down the alcohol in your liver. So if there's no alcohol in your blood, you don't need to spend energy and resources in making alcohol dehydrogenase. So the gene is turned on only when alcohol is sensed in your blood, which is, you know, a whole nother process determining the body determining when there's certain molecules in your blood, but it can be turned on and off. There's a lot that goes into gene regulation, but I at least want you guys to understand that genes are not always on. They're not always being transcribed and translated at all times. It depends on what the body needs. And that's it. So any questions about any of that material? Anyone on Zoom have any questions?
Go back to the last slide for a second.
Go back to the previous slide? Yeah. That one?
Yes, ma'am.
Okay. So what I'm going to do now, do you have questions about that, Braden? Sorry, or do you just want to see it again?
I just need to see it again.
Okay. What I'm going to do now is kind of give you an overview of what to expect with the exam. Oh, before I get into that, I actually put you guys on Canvas. I put you into disease project groups. So your project groups. So you can communicate through Canvas if you want. You can share documents through that setup if you go into your group. And your outline isn't due until February 26th, so you still have some time, but you can at least delegate kind of who's doing what. I put up the information for that part of the project, so it's under the assignments tab. That way you can just upload one outline, and then the same grade will be applied to your whole group, basically, when I look at the outline. So not everyone has to submit their own outline. It'll be a group outline. I'll talk more about that as we get closer to when that's due, but at least start thinking about some of the information that you want to include and kind of how you want to delegate the different duties for that project. And then also chapter outlines very quickly. So I graded the chapter 3 and 4 outlines. I noticed some people, I think you're still using the OpenStax textbook because chapter 4 goes into tissues, and this was chapter 4 in the Saladin textbook, which is the official textbook for the course. So if you're still using OpenStax, make sure you get the Saladin textbook, because now the chapters are going to start really not lining up well, and they don't have the same information in them. So I didn't take off any points if you had that, but just an FYI, start using the class textbook. All right. So now, yeah, a general overview of what the exam is going to be like before we get into the review quiz. So we are going to, so you guys have to be split because there's not enough room in one room. We'll be up in the bio labs, which is Stewart 221 and 223. I'll send out a reminder email, but not the AMP lab. It's just a little bit further down the hall. So you guys will meet outside of those rooms, and I'll put half of you in each room basically, so there's enough space in between to allow for social distancing. So everyone will be here. So you guys on Zoom, you're going to be here in person on Friday for the exam. It'll be 45 multiple choice questions and then 5 fill in the blanks. So it's going to be 50 questions in essentially 50 minutes. So it's going to be, some people are going to have a hard time finishing. Some people will get done pretty quickly. That always happens, but just keep in mind, you don't have a ton of time to think about every individual question. So 45 multiple choice, 5 fill in the blanks. The fill in the blanks will have a word bank. What else? There will be some sort of applied type questions and then some more basic questions. And this Kahoot quiz hopefully will give you a good indicator of the types of questions I ask. Kahoot limits me on how many words I can put in a question. So some of the questions on the test will be a little bit longer, but yeah, I think that's all I was going to go over. Any questions about what's on there or any of the material or the process of the exam? All right.
What do you want us to do if we're not going to be there?
If you're not going to be there, why would you not be here?
We have a game in Mississippi Friday.
Oh, you should definitely email me and we'll set up a makeup time.
Okay.
Yeah, if you ever can't be here for an exam, you should email me as soon as you know, and then we can set up a makeup time. So I have some people already making it up the next week. Yeah, so shoot me an email. And then let's see here. Yeah, if you have extended time, let's see. So I emailed everyone who has extended time. I'll get back to you and answer that in just a second, or after I go up to my office. Is there anything specific we should spend time studying to prepare? I'm basically going to divide the questions up fairly equally as to how long we spent on them in the chapters in class. So there will be slightly more from chemistry, the chapter 2, and then chapter 3, the cells. We spent a good amount of time on those. So I think I have like 9 questions devoted to the first chapter, so like the intro stuff, and then 8 devoted to this chapter, and then I think 16 and 17 somewhere in there for the middle 2 chapters, if that helps. So I try to space it out or divide it based on how long we've spent in class on that stuff. Any other questions? Yeah, you should definitely learn terms and definitions for the exams. Some of them I'll just ask kind of straightforward definition, like what does this definition describe? Other times it'll be more applied. So I'll show you some examples on the Kahoot exam. So yeah, look at the study guide. So the study guide should give you a pretty good idea. It's on Canvas under module, I think the Unit 1 module. That should give you a pretty good idea of the topics to study. So yeah, you're gonna wanna know terms. There's a lot of memorization in AMP. So you wanna memorize like the different parts of the body, et cetera. And then also be able to apply some of that to situations. Any other questions? All right, so we're gonna do a Kahoot review quiz. And I'm gonna, oh, I already posted this actually. I posted the link to the quiz on Canvas, so you should have access to it. If you wanna go back and look at the answers, et cetera. So for you guys on Zoom, I'm just gonna, like I did before, turn off the sound. And then just put you guys in front of the screen so you can see the questions, since I can't run it on 2 computers at once. You can join with whatever name you want. I don't really care. I'm not keeping track. This is more just to give you guys an idea of the types of questions I'll ask and to give you guys an idea of areas where you really need to focus and don't really understand certain concepts or haven't studied them. I will say don't leave this until the last minute. Start, like I've said from the beginning, start reviewing your notes now and studying now. It's a lot of material. So we probably won't have time to get through this whole quiz, but like I said, the link is on Canvas, so you can go back to it as a study tool. All right, so I'm going to start it. If you haven't joined yet, the info will be at the bottom of the screen, so you should still be able to join if you haven't quite yet. Okay, so an analogy to start it off. Anatomy versus physiology. All right, so yeah, anatomy is the structure aspect, physiology is the function. So anatomy is what muscle is this? Physiology is how does this muscle contract, basically. All right, what type of reaction is shown here? All right, it is decomposition. So hydrolysis was another option. So hydrolysis does break down the polymers, but I'm not showing any water being used here. So it wouldn't be hydrolysis or water being split basically. So it wouldn't be hydrolysis. And I probably I mean, thinking about it, I probably wouldn't give you hydrolysis as an actual option because that could be a little bit confusing. So I try to think about, and these are useful for me too, try to think about any possibility that could also be an answer. So decomposition technically is the right answer here. HCl, hydrochloric acid, is being broken down into its component ions. So it's decomposing. Synthesis would be the option or the opposite. If H plus Cl were formed into HCl, that's synthesis. And exchange, I'm not going to ask you about because we didn't really talk about it that much. All right. What is a chain of more than 50 amino acids called? Wow, that was pretty evenly split. Amino acids are the monomer for proteins. Amino acids make proteins. So you could eliminate, assuming you know these aren't proteins, you could eliminate all those others. Lipids don't really have a monomer. DNA, the monomer is a nucleotide. You want to know the monomer and the polymer name for each of those 4 macromolecule categories. Lipids don't really have a monomer, formal monomer. A polysaccharide is a carbohydrate. So its monomer would be a monosaccharide. So those are easy to get confused. So definitely spend time on learning the monomers and the polymers for each of those 4 biological macromolecules. All right. Which of the following is an example of a suspension? So this goes back to the mixtures topic we covered. Nice. Yep. So blood cells and plasma are the suspension here. So a suspension is something that will separate over time. And this is the exact example I gave you of a suspension in class. So blood cells are heavy. They'll settle out of the plasma. That is a suspension. Milk is a colloid. And then the other type of mixture we talked about was a solution. So these 2 are solutions. They basically don't scatter light at all. You can't even tell anything's really dissolved in the solvent. Milk, colloids are usually cloudy looking, so that's a good example of that, but they don't settle out over time. Blood cells and plasma do settle out over time, and you can't really see through like a test tube of blood. That makes it a suspension. Okay, so this is more of an applied type question. All right, that's kind of a tough one because first off, you have to know that I kind of gave you an extra hint here. So a lot of energy. You have to know what organelle provides the energy and make that association, which would be the mitochondria. So the mitochondria are the little energy centers of the cell. Muscles that frequently contract require a lot of energy. And so they have a lot of mitochondria. So that is an applied type question. Those are the tougher questions definitely on the exam. You have to know that mitochondria make the energy and then make that link to the muscle question. Body temperature regulation is an example of a what? Some kind of feedback mechanism. All right, it is a negative feedback mechanism. So you guys were equally split between negative and positive. Those were the only two types of feedback mechanisms we mentioned. So I'm glad you didn't put neutral or opposite. So negative feedback, the body temperature example is one that I focused on specifically in lecture. So this is where as your body temperature, say, increases, your body tries to bring it back down in the opposite direction. That makes it negative. So the reaction is opposite of the stimulus. So the stimulus is the body temperature going up, and your body is reacting by trying to bring it back down, say if you have a fever. A positive feedback mechanism, if we're talking about body temperature, which is not regulated in that way, would be if your body temperature is increasing, then your body just makes it increase more. So it kind of amplifies that response or that stimulus. So that is not what happens, thankfully, with our body temperature. So definitely go look at the different types of homeostasis and those positive and negative feedback mechanisms. All right, more homeostasis. In a homeostatic mechanism, what senses changes in the body? Yay. Okay. So yeah, the sensor or the receptor. So I'll give you both of those terms because they're both used. The sensor senses the changes in the body. So it senses the change and then it sends that information to the integrating or control center, which is usually your brain. It makes the decision about what to do. And then the effector is what carries out that action, whatever needs to happen. So for body temperature, for example, your skin would sense that it's getting hot, for example, and then your brain would say, okay, if your finger is getting hot because you're touching a stove or something, it will send a message to your muscles in your arm and your finger to say, pull your finger back. So those are the three parts of the homeostatic mechanism. Multiple tissues working together form a what? Nice, good. So multiple tissues make an organ. That goes back to the hierarchy of complexity in our body, starting with an atom. So make sure you know that trajectory going from an atom to a molecule, to a cell, to a tissue, to an organ, etc. Know what comes next and previously if I give you one of those options. All right, let's do one more. It's almost time to go. The following organ system plays a large role in processing sensory information. Nice, the nervous system. So you want to know the main functions of each of those organ systems. The nervous system, primarily our brain, is what makes decisions or processes information and makes decisions for what our body is going to do. All right, we are going to stop there. You can play the rest of this or just look at the questions as a sort of additional study guide as to what is on Canvas already. Email me with any questions you have or come by. It won't be on campus tomorrow, but I'll be around for all of today and then Friday morning. So I'll send an email reminder where to meet, all that kind of stuff. But yeah, study and don't leave it until Thursday night, please.